imagine that if I say that, let's say there's questions on this review, that some of them will, versions of them might appear on the final. It's not going to be exactly word for word. We understand about that? Okay. I have students that don't seem to understand that, like, what that means. The same ones that get frustrated that when I give the study sheet, they were like, I memorized the words basically on the study sheet and didn't pass the test. We're like, that you don't understand what understanding means. So, but yeah, so I'm just saying, it's the same student that I got this email this morning that the student emailed me. And like I said, I had written the final and accidentally put the wrong date in for the due date on, I wrote it in like my online section or whatever. I start in one, then I just copy it over. I wrote it in there, and when I had like published it as like an assignment or whatever, because I had put March, it populated everybody's results as 0 because nobody had taken it in March, and it's late, whatever. So that happens. She emailed me this morning and she was like, I had an A and now I have a D. And she's like, I thought that the final wasn't until day. And I don't know how this could happen. I thought did I do something wrong? And I'm like, I don't know if you're saying this to save face or if you actually believe that I'm just going to give people zeros on the final because I don't like you or I wasn't paying attention, or clearly this was a mistake. Like, anyways, those are the types of people that also I need to clarify that when I say, and you guys are in here, so I can say, and you're here and you showed up, the questions that we're having on here, many of them will be on the final in some form. Some of them literally, some of them, a different version of the question, maybe the opposite or whatever, but you guys get it. So I feel like that's pretty useful study material. That's something that I don't know that, was really necessary because it does just cover the same study sheets that we've been using. So it's not, and of course there's going to still be questions on there that weren't from here. I just want to be clear about that too. Like, it's not all from the Kahoot, it's some of it from the Kahoot. So, but hey, if you go practice the Kahoot, at least then it's something. So, it was just crazy. All right, we'll go ahead and do the classic mode. It seems to be what people prefer for now anyways. If we'd had more people, then probably would have been funner to do one of the other groups, but the other games, but it is what it is. Unless there's some other one. I wish there was a way for me to like try collaborate. It's a crew, so it's group based. I'm just looking through them really quick because I haven't actually looked at these. Team, kingdoms. I don't know what's happening in this one. Whatever, let's just stick with what we've been doing. I want the ones that don't have the timers to them. All right.
I figured they'd let you adjust that when you make it.
Huh?
I figured they'd let you adjust that when you make it. No?
Maybe some of them, yes, some of them no. Like, you can adjust, it'll give you like a time limit and it'll go for 10 minutes and then, the winner after 10 minutes. But I want to do something that's just like we go through all the questions for sure, which it should take about the right amount of time. I was reading this book. I'm just still trying so hard to get better learning. You guys are moving through the next semester, right? Reading the book, and it just starts out like one of the first chapters. Like, students will turn in, their first exam or their first quiz or their first essay or whatever, and they'll be so confident that they did so well on it. And then you go and you grade it, and as faculty, you're thinking, wow, what were they thinking? And then the student will get the grade back of like the C or the D, and they're like, what happened? Like, I felt so good about this and whatever. Yeah, tell me that isn't like exactly what happened. And so just so you guys know, I'm reading books about these are situations happening across the nation. And I'm trying to figure out a better way to approach things so that at least I got some good from my students. I'm trying. I really am. So if there's anything that you feel like worked particularly well for you guys, whether it is just Kahoot, it's as simple as that, or whatever. And if you guys haven't already done your eval, write that in there. And if you still have other things to say, you can always email me or even just, pass an anonymous note on in the science department and whatever. So, because I do want to know, I just like genuinely want to do better. So I'm always trying. I know everybody is smart enough to learn. It's just a matter of finding out how to help you learn. All right, so we're going to talk about units 1 and 2. This is basically what microbes are, how they function. So unit 1, remember this is going to be chapter 1, the basic intro stuff. Chapter 3, the tools of micro. And then we went back to chapter 2, chemistry. And then 4, 5, and 7 were bacteria, eukaryotes, and the viruses. That's what we'll go over. All right, microbiology. What is it the study of? All right, good job, guys. This is in fact going to be individual organisms that are too small to be seen with the naked eye. Most of this stuff hopefully really is going to be reviewed. So disease-causing agents are made of cells. Is that true or false? I like that. The first word is probably the most important one, right? The all. So no, because viruses are not made of cells and neither are prions. So we can rule those out. All right, what is the correct term for disease-causing microorganisms? Ready? This is going to be pathogen, just another vocab term, basically. We've used it enough in this semester now, it's not really anything new. If you're sick with a communicable disease, what's one thing that you would know for sure out of the choices? All right, we know that it's caused by a germ, and there's a term for this, right? It's called germ theory. So germ theory says that if you have a communicable disease, that it is going to be caused by something, right? It didn't come out of spontaneous generation. The reason that we wouldn't go with obligate parasite is simply because, sometimes we get sick with things that aren't always disease-causing organisms. And we can't, narrow it down to bacteria or a virus necessarily, but we do know it has to be something. It could be protozoa, it could be fungal. All right, the correct way to refer to the cause of the plague is Yersinia pestis. But what system dictates how to write that? Yep, binomial nomenclature. I mean, binomial being two name way of naming for nomenclature. So. All right, in taxonomy, kingdom is larger or more general than domain. All right, that one's going to be false. Remember, there's domain, and that's going to include, what are the domains? That's going to be bacteria, that's going to be eukarya, and it's going to be archaea. There's only 3 that fall into that. And then we have kingdom, phylum, class, order, family, genus, species under that. All right, inoculate, incubate, inspect. It's supposed to say identify. What is the missing 5I? All right, that's gonna be isolate. Good job. Pretty easy now that we know what we're doing with our bacteria. All right, what type of media allows only certain kinds of microbes to grow and inhibits the growth of others simultaneously? All right, perfect. That's going to be selective. Remember, differential is going to have color changes that tell you the difference. You can have both going on at the same time. If you have a growth factor added in, it's going to be called enriched. Right, the ocular lens of the microscope generates the real image. Is that true or is it false? This was on the lab exam, right? We all knew that was the ocular lens. I'm hoping everybody knew that was the ocular lens. Some people probably missed this. Well, I have it up on the board, don't I? I actually went over it pretty well with you guys. Not too bad yet. I'm not going to lie, like the other classes didn't get that sort of tutoring about the reminder there. So, right, that's going to generate the virtual. So the real image is at that actual objective lens, and then it travels up through some mirrors and lenses to get to the eyepiece where we have the virtual image of the optic lens. Because it's been through a couple of lenses and mirrors, it's not the real image anymore. All right, if the objective lens is 40 and the ocular lens is 10, what is the total magnification? I very basically switched this one up simply because we're so used to seeing the 10 and the 100, because that's what we always have the microscope set on. So not anything crazy there. Oh. What is the purpose of oil immersion? I feel like the question on the test is almost exactly this. And I actually didn't do that on purpose. I was just writing the test and I saw that this was one that I had dragged over from one of my other banks. And I was like, oh, okay, that works out perfectly. So light scatter, we're gonna reduce light scatter. Since the oil and the glass have similar refraction. All right, negative stain stains the background because it is repelled by the cell surface. Is that how that works? All right, so this one is actually true. The negative stain has a negative charge to it, and the surface of your cells also have a negative charge from those phosphate groups and the phospholipids. So they repel one another, and therefore the stain will stain the background. What type of chemical bond involves transferring an electron from one atom to the other? So focus on that fact that we're transferring. All right, excellent. So this can be ionic. What I always think of whenever I think of ionic bonds is the sodium and the chloride. You got the sodium, that's the positive, and the chloride, that's the negative. It's negative, the chloride is, because it stole the electron from the sodium. So that was that transfer. It's not the same as sharing electrons, which is what we see in covalent bonds. They're actually sharing, sometimes unevenly, but still sharing. Carbon dioxide is an organic molecule. Is that true or false? All right, so this one is going to be false. Just because it has a carbon in it doesn't make it organic, right? You have to have a carbon-hydrogen backbone, essentially making up the bulk of the molecule. Olive oil is a great example of an everyday hydrophilic molecule. True or false? All righty, this doesn't surprise me at all. I figured it would be that the case. Hydrophilic, phil being love, so water loving, but oil doesn't mix well with water, right? So it's hydrophobic, water fearing, just like an old yeller. Which pH is the most acidic? Okay, good job. I'm glad that you guys got that. So even though we typically think of 7 as being a neutral, it is the most acidic in the list for sure. So good job with that. Another question about pH. Which pH would have the highest hydrogen concentration? Alrighty. So this one is also not surprising because I'm glad that at least like we're at the ends. But two, because it's the most acidic. Remember, even though two is the lowest number, it's a negative logarithmic scale. And what that means is the more hydrogen it is, the lower the pH number. So hydrogens and how much hydrogens are there, hydrogen ions anyways, dictates how acid it is. Which level of protein structure features alpha helices and beta sheets? All right, so this is going to be the secondary level. Just to remind everybody, primary is going to be the amino acid sequence. Secondary, we're going to get basic structure of alpha helices and beta sheets from the reaction on the surface of those amino acids. Then the tertiary is starting to have interactions between those bigger groups, between one another in that one polypeptide. And then quaternary is bringing multiple polypeptides together to create functioning units. All right, true or false, bacteria and archaea are always single cell. All right, so this one is actually true. They don't form multicellular organisms, right? So they are always going to be single cell. They can form biofilms and things like that, but because each cell can still exist separately as one single cell on its own, that's why they're still considered to be single cell. For example, none of the cells in your body can do that. They can't exist and like multiply and, you know, live their life away from the body. They have to be part of the body. So bacteria and archaea, single cell. What cellular arrangement is featured below? All right, so this is strepto. Strepto meaning we're forming chains. So good job there. All right, which flagellar arrangement is shown in the image? If you can't see it very well, there's one coming off here and then one coming off on the bottom. How we're looking at that. So yeah, that's going to be amphitricus. So that's going to be both. Amphi is always both. Gram-positive bacteria have a thick outer membrane. Is that true or false? All right, so this one's gonna be false. Excuse me. This is false because they don't have a thick outer membrane. They have a thick outer wall. So peptidoglycan wall on the outside for the gram-positives. Gram-negatives have an outer membrane, but I wouldn't say it's a thick outer membrane. All bacteria are capable of making endospores if nutrients are scarce. Is that true or false? All bacteria are capable of making endospores when the nutrients are scarce enough. Right. Thank you. I was worried. Right. So this is absolutely correct. You guys did better than my last class on that question, at least. So definitely false, right? That's going to be only Bacillus and Clostridium. So which group would Neisseria meningitidis, gram stain is going to be pictured, fall into? So this is pink, or you could see it as red. So which group would you put it in? Alrighty. So good job. You guys also did better than my last group. This is the gracillicutes. So just real quick, we'll run over what these are and why. So like I say, when I have all four choices, and I'll only ever give you the four choices that are like these four, so you don't have to like parse out someone that sounds similar. But Mendoza Cutes reminds me of Joey Mendoza. When I was in middle school, he was my boyfriend. That was a long time, and that's archaic to me. So old, archaic. Archaic fall into this category. The Firmicutes, they're firm because they have that thick peptidoglycan cell wall. So that's the gram positives. This is the next one that I would go to between the tenericutes and the gracilicutes because G goes with gram, and which gram is left? The gram negatives. So that's why I pop them in here. That's how I remember it. And then the tenericutes is the ones that don't have a cell wall at all. So that's like mycoplasma. Yeah. So that's how I remember. That's how I get to that conclusion. All right. True or false? Some eukaryotes can use cilia for locomotion or motility. Alrighty. So this one is going to be true. I mean, yeah, good job. We have cilia that we can use to move around. We have flagella, just like bacteria do. But they just operate differently, right? So remember, for the eukaryotes, they use a whip-like motion. The bacteria have the rotation like a motor, but they both have flagella. But these guys can use cilia to move around. They can also use something else to move around. Some groups can use pseudopods, so the fake feet, and that's generally how your like macrophages and stuff are going to get around. So, all right. What is the function of the mitochondria? I don't know what it is about the mitochondria, even though it's like one of those words that is initially kind of a difficult word. Most people remember about what it does. One of my students in my last section was talking about how he remembered it from like high school or something. It's just one of those things that sticks. So which step of mitosis is pictured? This is a difficult one. I won't have a picture like this on the test, but if you can tell me what it is, at least you can understand the concept of what happens in mitosis. I would at the very least probably know what the different steps are in mitosis. So PMAT, right? So be aware of that and what each letter stands for. But that was anaphase. That's because for PMAT, P, prophase, that's when everything is just getting set up. We are coiling up our chromatin so that we can have our chromosomes, nice and consolidated and whatever, as well as making like the parts that we need for the whole process. In M, metaphase, we have lining up of the actual chromosomes in a line and attaching to the spindle fibers. And then in anaphase, we're pulling those chromosomes apart. In telophase, we are separating the new daughter cells. So that's basically what's going on in those steps. All right, what is the term for the overall structure of an entire fungal mass? The whole big thing. All right, so the word for the big whole overall structure of the fungal mass is going to be mycelium. Hyphae, to be clear, that's like when you look at mold, like typical mold, the fuzzy looking kind, right? Those little individual hairs that are coming off of it, each one is the hyphae. And then the sporangiophore and the conidiophore, they are reproductive, asexual reproductive structures. We learned about the conidiophore whenever we talked about Aspergillus and penicillium in our lab stuff. That was the conidiophores for those. So those are the open ones. The sporangiophore are the closed ones. And that's where the asexual spores come from. Yeah. I'm glad we didn't spend a lot of time on the fungi because I just don't know a lot about them and it's weird. All right, so what is the term for free living metabolically active forms of the protozoa? So when we're talking about bacteria, the free living metabolically active ones are vegetative. When they are dormant and they're just hiding out waiting for better conditions, in bacteria, that is endospore. Now, the correlating terms for the protozoa, we have trophozoite is the same thing as vegetative basically, and cyst is the same thing basically as the endospore. So that's how those work together. Just always remember vegetative and endospore, always bacteria. Right? True or false? Helminths are animals. We're talking about the kingdom Animalia here. All right, so this one's true. They do fall into kingdom Animalia. A lot of people get sidetracked because when we talk about kingdom Animalia, that they automatically think it's got to be mammals. Don't forget, I mean, not only do reptiles fall into that, and that's a whole other thing, but like even insects fall into kingdom Animalia. So, but yeah, so do these guys, the helminths. Select the types of genomes that viruses have been found to have. See if you can pick them all. All right. So the answer for this was just all of them. Surprise. So it can have any single-stranded or double-stranded DNA or RNA. Remember, it can be positive or negative sense as well as their single strands. So, all righty, anything pretty much. True or false? All viruses have capsids. Righty. So this one's actually true. It tends to be that when people are selecting false for this one, they're not really thinking of capsids. If they think of capsids, what they're thinking of typically is the envelope. So remember the envelope is the lipid bilayer that they get from the host cell when they're budding out, and they don't all have that. So you're right about that concept there. Thinking isn't in the wrong direction. But they do all have capsids. The capsid is just that protein coating around the nucleic acid. That's it. So they all have that. The bare minimum is capsid and nucleic acid. All right, where does that viral envelope come from? If it did have an envelope, that lipid bilayer coating, where does it come from? All right, perfect. Good job, good understanding of what's going on there. All right. The lytic phase is the phase when bacteriophages actively multiply until they burst out of the host cell. Is that true? I loved this. This was like my favorite find of this whole thing. It's so stupid, but it is my favorite. Alrighty, so this one's true in bacteria. Their viruses will come in and inject their DNA in and then multiply up their virus progeny and then burst out, right? So we know that at least is that the lytic phase? Yeah, that's the lytic phase. There's one other phase they can go through, and that is lysogenic, right? So that's gonna be when they incorporate their DNA into the genome and basically hide out in the bacterial cell that way. And they can multiply when the bacteria multiply that way. What is the term for viruses that can cause cancer? I think we're going to find out more and more as medicine does more research that a lot of cancers were probably originally associated with some sort of virus. Whether that's something that you end up carrying as genes now, which I don't know if you guys know, this is something like, I think over half, it might not be quite half, but it's at least 30% of your DNA is like virus. And it's just hanging out like just old. They don't know if it does anything or if it ever did or if it really was a virus at some point. But that stuff I feel like is terrifying and cool at the same time. All right, so oncovirus, just like oncology. What kind of genome do prions have? This is a trick question. Is it related to the viruses? Is it, I don't know what SSRB little a is, but it's down there. So probably thing I wasn't paying attention to what I was typing. Yeah, obviously they don't have genomes. They are just protein that's misfolded. All right, we'll move on to unit 2 now, which I know is the heavier material. I like it to go over unit 1, but right before it, though, because it does help kind of refresh our memories about those other things. When we go into this stuff, it's a little bit easier. But we'll talk about the genetics, genetic engineering, microbial nutrition, ecology, metabolism, disease, and epidemiology. All right, Jenny has brown hair, but she has one brown gene from mom and one blonde gene from dad. Her brown hair color is what? Alrighty, so this is going to be phenotype. Remember, her genotype will be the brown gene and the blonde gene. But since what we see expressed is the brown hair, that's the phenotype. The phenotype is what we see expressed. I'm waiting until after the finals to dye my hair again. Which one term describes DNA replication best? I feel like the gray is showing and I'm not a fan of it. I don't dye my hair very often. I dye it like once every like 4 months or something. But alrighty. So the best term to describe DNA replication out of these choices, right, is going to be semi-conservative. That means, of course, one strand will be the old, and then we make the new one, the copy, and so it's old and new every time that you make a copy. Not nuclear, and nobody chose that one anyways, but even though it happens in the nucleus in eukaryotes, it doesn't happen in a nucleus in bacteria, right? Non-canonical, we know what that means, it's not what's happening. And then transcription. So that was a popular choice in my last section as well. Remember that transcription is going to be DNA to RNA. So when we're talking about DNA replication, that's DNA to DNA. So it's not going to be transcription for that one. Oh my gosh. There it is. All right. The building blocks of DNA and RNA are known as nucleotides and consist of phosphate, pentose, and nitrogenous base. Is that right? Look at that. It's terrifying. It looks like a monster. I think it's supposed to be RNA polymerase. It's terrifying. So this one, this is true. We have DNA and RNA. They're both made of nucleotides, and a nucleotide is a phosphate, a pentose, which is just a 5-carbon sugar. We talk about DNA, that's deoxyribose, that's that 5-carbon sugar, or RNA, which is ribose, which is that 5-carbon sugar. The nitrogenous base just means what makes it the A, the T, the C, the G, or the U in the case of RNA. ID, true or false? Exons are non-coding regions in eukaryotic DNA that must be excised to make a usable mRNA transcript. Is that right? Every time I talk about exons, I think about the exon Valdez. Okay, so this one's false. Majority got this. So the exons are the coding regions, but not the non-coding. So the non-coding have their own name, introns. So those are the guys that introns we cut out and we splice the exons together. And splicing is what we call it. The next question asks more about it. All right, no, it didn't. So what are codons used for? What are codons? Right, green and red are typically the popular choices here. Most of you guys lean towards the red, and I'll tell you why red is wrong. Because codons aren't found on DNA, they're found on messenger RNA. Okay, when they're on DNA, they are called triplets. That's all they're called. So that's the giveaway there. A little bit tricky on that one. These won't be choices on the exam, but at least gets you thinking about the difference between that. So yeah. So yeah, the green one is what's correct. 3-nucleotide code. That part's correct, absolutely. And it tells which amino acid to attach next, but it's going to always be on RNA, the messenger RNA. All right, if we have a codon, ACA, and that gives you threonine, a mutation change it to AGA, now we have arginine. What type of mutation is this? So we've changed one amino acid for a different amino acid with this one nucleotide change. Right. Yes, you guys got this one. This is a missense. Remember, that's going to be anytime you have amino acid change happening. If you have a nucleotide mutation that causes it to turn into a stop codon, that's nonsense. And then if it just is the same amino acid because the overlapping code ends up being the same, then that's silent. And then, of course, fun sense is completely made up. All right, what types of DNA sequences do restriction endonucleases tend to cut at? Alrighty, so it's going to be palindromes. These are the same forward on one strand as they are on the backward part on the other strand. So they recognize those sequences and create, what kind of ends do they create? Sticky ends, right? So that's useful for inserting genes into things like plasmids and whatever. All right, what is the purpose of PCR? We talk about it a lot, but what does it do? Alrighty, PCR basically just replicates DNA, makes copies of DNA sequence. And you have to know the DNA sequence that you're making copies of. Right. All right, second step of PCR is anneal. What is being annealed? So anneal is to stick something together. The first step is denature. Then we have anneal. Then we have elongate, just to remind you. So what is happening with anneal? All right, perfect. So much you guys getting this one. This is primers. Those primers that we're adding in so we can target a specific sequence, they're going to anneal to the single strand DNA template. Additionally, the reason that that happens and the DNA doesn't just stick back to its other copy is because we have that in excess, the primers. There's going to be so much more primer than the other DNA strand that they'll bind instead of the other DNA strand. What else do I want to say about the primers? We have to have primers there also to build off from because the polymerase doesn't add nucleotides to nothing. It has to add it on to something. Kind of like adding train cars onto other train cars. In DNA gel electrophoresis, what is causing the DNA fragments to separate by size? So it's actually pushing the fragments apart or making them move in the gel. Alrighty, so this is actually electrical current. That electrical current moving through the gel causes that negatively charged DNA, remember they have phosphates there, it's got that negative charge, move to the positive end because negative will be attracted to positive. So that's what causes it to move. All right. BT corn produces proteins from bacteria toxic to certain insects, but not harmful to humans. What does that make it? What is the corn then? It has these proteins from bacteria, make them toxic to insects. There's more than one answer. All right, so this one's going to be transgenic and genetically modified. These are the same thing. So FYI, and this is real. And I actually was reading online about how like about 95% of all soy products are GMO now. So probably odds are if you're having any sort of soy product, soy milk or soy sauce or tofu or anything like that, that it's probably GMO. It's the case with corn in general as well, but it just depends on what you're buying it from. Somatic gene therapy can be passed down to future generations. Is that true or false? So obviously there's one key word here that's somatic gene therapy. That word means. All right, so this one's false. So the one that can be passed down to future generations is called germline. So yeah. We've seen the terms, we recognize them. Now it's about bringing them back out of the depths of memory. What is the term for an organism that gets its carbon from inorganic sources and its energy from the sun? Okay, so the term here is going to be photoautotroph. Photo from the sun, obviously. Auto, they're getting their energy, they're getting the carbon and turning it into organic compounds they can use, but they're doing that themselves, auto, so yeah. Or what is the term for a nutrient that must be provided in the diet because an organism can't make it for itself? Right, so this is going to be growth factor. A lot of the vitamins that we get in our food are that for us. So like vitamin C, like if you don't get enough of it, you get scurvy. Vitamin B12, a lot of people don't absorb it very well in their gut, which is probably just due to unhealthy gut biome if we're being honest with ourselves. But I'm one of those people, I'm not judging. So yeah, we'll have to supplement. And I use like sublingual tablets, but a lot of people, it can get bad enough you have to get the shots. So I did it first. What type of solution could have a typical cell swell up and burst like a balloon? Almost this exact question is on the exam. You take a typical cell, put it into a solution, and it swells up big and then just bursts. So that's all right. Most people do want to put hypertonic, but it's actually hypotonic. And there's not an eraser. I want to draw it on the floor so you can see it. Are over there. So really quick, so we can go over this stuff. We have, if you put cells into solution, of course, if it's the same on the outside as the inside, the term for that is isotonic, right? That's fine. So if you have a cell, here's cell A, and it's got some stuff in it, ribosomes, salt, and it's not all pure water, right? Got some stuff. Same kind of cell down here. Okay. In scenario number 1, up here, we're going to put it in a solution where there's a lot of stuff on the outside. Man, this cell went swimming in stench cone, right? So it's in the stench cone wildlife preserve. It's in Overhaul Surf. Maybe in the ocean, if we're talking about actual salt, But either way, which one has a higher concentration of water? The cell, right? It has more water concentration. Water is what can cross over. This other crap can't. So what it does is this has more water, this has less water, so it donates water from inside and tries to push it outside to try to make the concentration equal. So when it pushes water out, it's going to shrink down, right? This will shrink because this one on the outside out here, this is hypertonic, okay? All right, so if we look at this one down here, and we've got stuff in the cell down here, we've got maybe, I don't know, a little bit, almost pure water, let's say, okay? which has the higher concentration of water now? The outside, right? So water will rush in from outside to try to water down to the inside so that eventually it'll be the same. That's what the goal is anyways. That's what it thinks the goal is. And trying to equalize those gradients. So this situation, this solution out here is called hypotonic. And then that will cause the cell to expand and then eventually burst if it gets too much water moving in. When we have cell walls, or we have an extra membrane like bacteria do, either way, can help protect from one or the other, depending on the organism and if it's salt versus something else or whatever. But yeah, they have ways to protect, but clearly we can see the water moving in is what will make it swell or expand. That makes sense? Cool. It's been a while since we touched this stuff. All right, pathogens and microbes of our biome thrive at temperatures near or around room and body temperature. So what does that make them? Mesophiles is the word for that. So it's like at the middle temperatures, what we consider not high and not low. What role do bacteria play in the nitrogen cycle? So don't get too heavy into this, but what roughly role do they play? And how nitrogen is recycled in our not atmosphere necessarily, but just environment. That's the word I'm looking for. Ready. So they convert it to a form that plants can use. That's what they primarily are going to do for the nitrogen in our ecosystem. Taking it into forms from like nitrate and stuff like that into more usable forms like ammonia-based compounds for the plants to use. Without them, the plants can't get the nitrogen that they need from the soil. So what are coliforms? What does that term mean? Alrighty, so these are indicator bacteria. Yeah, that's absolutely correct. They're basically going to show you whether if these guys can get through the filtration system, basically, then the pathogens can too. Even if you want to say we could just test for the pathogens, wouldn't you rather like plug the leak before the pathogen gets through it? So that's the idea, is we can see normal E. coli and stuff like that in there before we have Shigella, before we have the actual, S-tech E. coli and stuff getting through. And we would want to know sooner rather than later. All right, what is the term that refers to chemical reactions in the body involving breaking down molecules into smaller products? My security system will like alert me whenever there's like a person at my door and all that stuff. Normal, that's normal stuff. It's a Ring camera, but like I have other cameras around and like it'll tell me if there's motion at certain places, like it doesn't recognize it as a person, but there's something moving that wouldn't other be. And I'm like, I'm always worried. I'm like, is there a monster? Like, is there ever going to be like a monster? They don't call it a person. It's something else. I don't know what's going on out that camera. Anyway, so catabolism, and the cat kind of gives it away, but that's kind of the point. Help remind you guys, catabolism, we're breaking bonds. We have destruction of molecules going on. So enzymes help overcome activation energy of starter to products. What is the correct term for starter? This is an actual animation of what it really does look like whenever proteins in your cell move large vesicles containing stuff around. Pretty crazy. Looks dopey, but it is accurate. Right. So we've got substrate. That's the term for that. And the substrate is going to be held by the enzyme in the active site. Now, if it needs some sort of other thing in order to work, then that would be the cofactor. And if that cofactor is an organic molecule, it's called a coenzyme. These are all words to be familiar with. All right, what does ATP stand for? Be careful with this one because there's some similarities. This is a demonstration of how ATP synthase works. This is our proton gradient down here, and it'll come through, and then that triggers it to be made into ATP. It's pretty cool, in my opinion. All right, so we've got, obviously, people were going to be choosing between the probably most prominently adenine triphosphate, can you say it? Adenine triphosphate and adenosine triphosphate. Adenosine is right. I'll tell you why. And I'm not going to put this on the test just so that we're clear. I'm not going to make you have to choose between those two because that is kind of ridiculous. But just so I can bring it up and remind us, adenosine, this means that it's a nucleotide. We've got the sugar added in. Otherwise, this is just a base with 3 phosphates. Okay, so this is the base with a sugar with 3 phosphates. That's all that means. So that's the difference there. It's more or less just asking us to recognize the term. But it won't be that tricky on the test. So in aerobic respiration, what are the 3 stages of energy production? Pick the 3 correct ones. All righty. So from start to finish, it would be glycolysis, then Krebs, then the electron transport chain, right? Gluconeogenesis is something that your body can do if it needs glucose to put into aerobic respiration. It can convert other molecules into glucose. We didn't really learn about that, but it is something that your body can do if it needed it to make more energy and didn't have glucose, which is the most effective thing to put in. The Calvin cycle, this is the dark reactions in photosynthesis. And then we have fermentation, which is like a whole other thing, right? So fermentation, that's going to be what organisms do when they don't have oxygen, which I think our next question is about fermentation. Not yet. Not yet. So what is the final electron acceptor in aerobic respiration? We said glycolysis, Krebs, electron transport chain. What's our final electron acceptor in all of that? All right, so this one's going to be oxygen. All those electrons shuttle through those cytochromes, and then all those protons, we've created the gradient. Protons move through ATP synthase to make ATP. Now we have a bunch of protons that are coming through. We need to maintain the gradient, so we need to get rid of those protons coming through. Plus, we have electrons that'll build up if we don't take care of them. The H pluses with the electrons to make them H's, with oxygen to make water to get rid of all of that crap. Okay, so that's oxygen. That's our final electron acceptor. All right, true or false, fermentation generates just as much energy per glucose on average as does aerobic respiration overall. All righty, so this one's false, right? So glycolysis, then Krebs, then electron transport chain. That's aerobic respiration. However, in fermentation, because we don't have oxygen to use as our final electron acceptor, we can't go through all of that. So we will go through glycolysis. We create an electron carrier, which we do need to get rid of that electron. So they use pyruvate, which is their waste product from glycolysis, right? So as a product of that reaction, we'll get lactic acid and or ethanol and or carbon dioxide, common products of fermentation. But that's all they do. And all they make in that is 2 ATP from 1 glucose molecule. Whereas typical cell will make 36 to 38. So eukaryotes make 36, bacteria make 38 ATP. Pretty impressive with aerobic respiration. So definitely most of that's happening in the electron transport chain, FYI. All right, based on what we've learned, which organism would be a true pathogen, an actual true pathogen? Only one of them. Okay, so this one's going to be Yersinia pestis, which is the cause of the plague. This wasn't meant to be any sort of trick. These are actual, like, spelled correctly, and these are the diseases associated with them. But Candida albicans, can you have that as part of your biome? Absolutely. So that doesn't always have to be a pathogen, right? So it's not a true pathogen. Staphylococcus aureus, it can be the same thing, right? It can be part of your normal biome, but it can also cause other diseases. And that is true of Streptococcus pneumoniae. We learn it as being the most common cause of community-acquired meningitis and pneumonia, but you can just have it harbored in like your nasal cavity naturally. So if these can be part of your normal biome, actually, if they're part of your biome, they're doing good for you. Even if they're just taking up your stadium seats to keep away the bad guys from taking hold in your body, even though they're still doing that, they're still doing good for you. So that means they're not always bad. But Yersinia pestis, you get that in your body, it's always a pathogen, right? So that's what a true pathogen is. Always bad. Should never be there. All right, virulence factor. What's the definition? What's the best fit for a definition anyways? Right, wonderful. So this is going to be something that increases pathogenicity and infectiousness, right? Anything that helps it infect a thing or whatever. So if we look at these other choices, in red, we have effects that can be seen in cells which have been infected by viruses. And those could potentially be considered a virulence factor, but that's not the definition of overall virulence factor. And that's kind of what we could apply to the blue and the green as well, if we're being honest. We need a big, more blanketing statement. None of those guys actually fit that bill overall, but they could qualify within that category. So which term refers to an agent that came from outside of the cell or the body? I don't know how I missed the crow on his shoulder the last time. He's got a crow on his shoulder. I don't know what that's about, but okay. I just thought it was funny. Anyway, so exogenous, yes. Exo, outside. These are terms we've heard a lot now. Right, could be more than one for this one. Which of the following are symptoms? You guys love these questions, right? Oh my gosh, I forgot about that. Okay, so we've got fatigue and headache because these are basically subjective. Lymphadenopathy, to be clear, I had a student ask me what it meant. It means swollen lymph nodes, but if you guys ever have a term on the test that's you're like, you don't really know what it means, and you don't recognize it as being something we directly went over as a term necessarily, or if you're just like, you really just don't know and you just want to ask anyways, it never hurts to ask. So you can always just ask if you're not sure. And I can always just say to you, can't tell you that one, that one you should know or something. But if it's lymphadenopathy, I probably would tell you, okay, that's swollen lymph nodes. Like, that's okay that you don't know that. I sometimes don't know what you don't know. So take it for granted. All right, in which phase of infection are the very first signs and symptoms seen? All right, so we've got all four of the stages of infection listed here. Incubation is whenever you have an introduction of the organism into the body. You haven't got symptoms yet because your body hasn't responded to it yet. It's just starting to get a foothold and grow. The next step will be prodromal. The prodrome or the prodromal phase, either way is correct. That's going to be when you see your very first signs and symptoms. Your immune system's just starting to mount a response to it and realize that something's wrong. The next step would be acute or infection phase. That's like the height of the infection. You've got full blown of that illness. The infection is really mounting up and your response ramping up. You'll have very probably the height of your symptoms and your signs as well. And then we have convalescence. You're clearing the pathogen, and now hopefully the signs and symptoms are easing back as a result of that. Okay, a fly flies out of the porta potty and lands on your food. If it's not infected, the fly isn't, but it's gross and you might get sick. So what is the fly in that? I don't know if you guys have seen this movie or not, but it's a movie that I have seen a couple of times and I don't watch it anymore because like it was traumatizing for me as a kid, like when I was younger, to see Jeff Goldblum turning into a fly. That's what it is, the fly. If you haven't seen it, that's what that movie was. But he gets into one of those like atom reassorter things. He's trying to have like teleporter or something is what he's trying to do or something. I don't really remember, but a fly gets in there with him and he becomes part of it. And at first it's fine, but then gradually over time, he like loses his teeth and like it's body horror stuff, right? So yeah, I don't really care for it. My husband's really into horror movies. That's just one I won't watch because it just like triggers the trauma. I watch horror movies. I love a horror movie, but like for some reason, the childhood trauma aspect, it doesn't. Mm-mm. So the fly in this one is mechanical vector. It's not itself being infected, but it is being exposed to whatever that thing is and carrying it around two things. We could best compare it in the list to the fomite. But fomites are non-living things that would be vector transmitters. But this is a living thing. Since it's not infected, it's not a biological vector, which is what is required for that. And an etiological agent is going to be whatever the bacteria would be that would be making you sick, salmonella or whatever. The actual pathogen would be the etiological agent. All right, we're getting there. We're almost there. We're almost there. So who was involved in identifying the source of cholera in the 1800s in London? You guys remember this guy? The picture is really dead giveaway, you can tell. But anyways, yes, it's John Snow. So John Snow was the guy that like figured out what water pump. There's the whole thing with the water pump, and people kept drinking out of it, and finally he just took the handle off of it to keep them from getting cholera. Father of epidemiology in a way. So multiple selection here could be more than one. Which term refers to the occurrence of a disease in a given time frame? So how many cases in a month? How many cases we've seen this week? Could be more than one, I'll remind you. Alrighty, so this one is incidence and morbidity. They mean the same thing, really. But if we look at prevalence, that's talking about the occurrence of the disease in that outbreak as a whole. We're not talking about in a month or something, how many cases have happened so far. That would be the prevalence in that population. Mortality, of course, is death rate. So incidence and morbidity for this, they're just the same thing. So a certain number of illnesses in a month or a week in a certain time frame. So that cholera outbreak in the 1800s, we're getting from that water pump, but it happened over several weeks, right? People one water pump, but people were revisiting and getting sick. So what kind of epidemic is that? The version of this question is on the test. I remember it. All right, so this is common source epidemic. The reason it's not point source will be like all at one time in one place all at once. So if somebody came in here and was just coughing up a storm with some new strain of COVID and we all got sick from it, that would be a point source epidemic, all got it at once. Since this was over a longer period of time, it's going to be common source. But they all went that same pump. All right, an outbreak that exceeds the expected prevalence, but which does not cross continents, is known as They're used to seeing a few cases of something every now and then, but now we've got suddenly hundreds or thousands, but we're not crossing continents. Or you could even say you've never seen this disease in your area, and now it's here. Right, so the correct term for this is going to be epidemic. Sporadic would be like one or two random cases that weren't really a true like outbreak. A pandemic is going to cross continents, and then endemic is something that is normally there in that it's just the normal amount of what you're used to seeing for that disease. All right. Could be more than one. What does it mean for a screening or diagnostic test to be highly sensitive? I hate these. The sensitive versus the specificity ones. They trip me up too. So I don't feel like it's just you. I have to think it through. All right. So there's two that qualify here, false negatives being less likely, and the test will trend towards identifying any possible positive. So think about it this way. If you're really sensitive, even just emotionally, let's say, right? Really, really sensitive, any little thing is going to trip you up. Yeah. So that's the idea here. You're really, really sensitive test, you will see any positive. You will find any of the positives. They'll really set you off. So therefore, you're not going to have false negatives as much because you're going to be picking up all the positives. You won't be calling them positive negative by accident because you're so sensitive. The other flip side of that coin is, of course, the specificity, recognizing true positives with minimal crossover, and having false positives being less likely. So that's the flip side of that. It's easier for me to remember the sensitivity and then just compare from there. Personally, that's what's easier for me. But specificity would be like saying, like, you can tell just because these people look very, very similar, you can tell the difference that they're not the same person, sort of thing, like telling that stuff apart. That would be specificity. No false positives. What is the term for how many people in an infected individual is likely to infect? Not in, but you're sick. How many people are you going to infect while you're sick? What's the term for that? All right, correct. That's going to be R naught. I'm glad we recognize that term. All right, so that's that. So that is units 1 and 2 complete. Do you guys have any questions about any of that material? I know, man, I'm at the end of it too. I'm like over it. So I completely get it. We'll have 3 and 4 next time. I feel like unit 2 is the heaviest of all of the units personally, but unit 3 is pretty heavy too with that immunology stuff. So if immunology stuff is the stuff you want to work on the most, be sure you go back and find some of the study session ones to refresh your memory about that if you need to. So anyways, I'll see you guys back on Thursday, same time, same place, and we'll do this again.